Hello, we'll be discussing an open source platform for quantitative interactomics. Its name is Life123, and it has a slogan. More on this later. The slogan is AI and big data aren't enough. We also need dynamical modeling. This video is an introductory, deeper dive. If you want a brief intro, there is also a 3-minute video, link in the description. Let's start with an overview of the Life123 project. It's an open source engine for quantitative reactomics, but what does it mean? It means that we perform dynamical modeling of biological systems, and the initial focus is towards eventually creating whole cell simulations. The platform consists of Python libraries, which allow us to do in-silico experiments. This project recently left the beta stage after a few years of development. So what's the motivation for doing all this? Well, there's a practical side and a conceptual side. The practical side is a simple one to state. About 90% of clinical drug development fails. That's appalling. And all our current AI and all our current big data, it seems they cannot do too much about it. So maybe we ought to be much more quantitative before we intervene. And then, moving on to the conceptual motivation, we encounter questions such as how does life emerge from a network of reactions, diffusion, macromolecules, barriers, membranes, and so on. Enough of a preface. Let's jump into the elephant in the room here. Let's consider a very simple network of hypothetical reactions. Chemical A reacts to produce X, which is our product of interest. And in a separate reaction, A produces B. B in turn produces X and C through separate reactions. And finally, in red here, C inhibits X through yet another reaction. This is all very schematic, of course. But now let's say hypothetically that reaction 5, the one shown in red, the inhibitory one, let's assume it's pretty insignificant in mice, but major in humans. So what's the net result? Let's say that our goal is to enhance X because, for example, the direct reaction from A is not enough to produce it. Our experiments in mice would show us that enhancing reaction 2, which produces B, which in turn produces X, would be a good thing because the side branch that leads to C and then inhibition of X is not significant in mice. So our experiment in mice concludes that's one way to increase the production of X, our goal. But then we move on to humans, and there the inhibition path is significant. So, lo and behold, there is a side effect. X does not get increased after all, maybe even decreased. Millions of dollars of development and clinical trials down the toilet. There you have it. This is just a contrived example, but even in a minimalist example like this one, it's pretty easy to come up with some scenario that could go in very different ways. And we don't know which way it goes because we're being qualitative. So that's what I call the tragedy of the qualitative. We have no clue about the actual evolution of a dynamical system. This screenshot is from reactome.org, an organization that builds a database of all the known interactions among molecules in humans, which is amazing. To me, it feels like parting the curtains of life and being afforded a peek at the inner workings of nature. But we still cannot predict what happens or how to change the physiology in a way that we want to change it. And now imagine if we had all this and we had numbers and a sophisticated dynamical simulator. Imagine what kind of power that could generate for us to understand and manipulate biology. So here we get to the fundamental point of this video, that in the path to the advancement of medicine, there's a lot of work being done on big data and AI, which is awesome. But at the same time, Dynamical modeling is getting short shrift. That's the premise here of this project. Now, going back to the nuts and bolts of what Life123 is in some detail. We said it's quantitative modeling for biological systems. In particular, the focus for now is for whole cells, initially prokaryotes, the simpler ones. And it's a software platform on which to perform in-silico experiments. Some of its features involve the ability to simulate networks of reactions, diffusion, membranes, and macromolecules, among other things. Life123 is open source, and it is integrative, extremely integrative. That's a key feature, many disciplines coming together. And it aims big. The idea is to be in a ready state for advances in computing power, as well as advances in knowledge of chemical reaction parameters, such as kinetic parameters. Another way to state it is that we want to do quantitative reactomics with spatial awareness, including compartments, transport, and diffusion. And what about the name, 123? The name is to do with simulations being available in one-dimensional scenarios, as well as 2D and 3D, and also in what you could call 0 dimensions, which would be uniform compartments, where everything is always perfectly stirred up. The fundamental idea is that work in lower dimensions paves the way in terms of being a stepping stone to get started more simply and quickly, and then proceeding on to higher dimensions. Equally important is to talk about what Life123 is not. And one thing it's definitely not, and it's often an assumption that many fall for, it's not molecular dynamics. It does not aim to compute what the kinetics of a particular reaction are. It assumes that the reaction rates, binding constants, diffusion rates, and so on are either known or estimated from measurements, or from AI models, or from molecular dynamics. And something else it's not just reaction diffusion as an end to itself. More of what Life123 is not. It's not a tool primarily for educational purposes, though that's a great side goal. It's not a computer game, it's not an art project, though it can generate beautiful images. And it's not mathematical exercise in dynamical systems. It's not cellular automata in the style of Wolfram. I don't have time to elaborate on that. It's not an exploration of hypothetical physics in 1 or 2 dimensions. Here, when we talk about 1 or 2 dimensions in Life123, we're talking about really idealized scenarios. 1D is like a long pipette, while 2D is like a petri dish. Let's talk about fundamental goals now. We aim to do detailed quantitative simulations of biological systems, and we would like to get deeper quantitative insight into biological cells and their interactions with environment, including drugs administered, and ultimately a deeper quantitative insight into human tissue, organ, and system physiology. It's a very integrative approach that could ultimately be conducive to insight at a body-wide level for health, aging, and lifespan. Those are the ultimate foundational goals. Other goals include the exploration of what is the minimalist essence of a lifelike dynamical system. In other words, what's the minimum you need for a dynamical system to have some kind of lifelike quality? Along those lines, that could be of potential interest to investigate paths for the emergence of life on Earth or on exoplanets. Finally, a fundamental goal of Life123 is to be a community effort that brings together people from multiple disciplines. So why open source, you may wonder? Well, if I had to use just 3 words, I would say long attention span. Just as a few examples, if you look at some of the familiar names in open source projects like Python, NumPy, and so on, their start dates go quite a bit far back. Open source projects live on, while projects at companies come and go as companies change their focus or departments lose their funding or companies go out of business. An example close to me is from another open source project I lead, brainannex.org. It started in 2015 and it has long outlived work projects at all of the several companies where I worked at. Open source is also something that's widely available to all researchers to use or contribute to without being shackled to any one company. I often see in the promotional literature of companies the term proprietary platform with the implicit implication that it's a wonderful thing they have. But to me, when I see those words, it has the opposite implication. To me, a proprietary platform is something that's at the mercy of that particular company. If that company shifts its focus or fails, then the project will likely be neglected or abandoned. And even if not neglected, it's still something that won't get any creative input of the rest of the world outside that company. So, proprietary platform is a big negative to me. Is Life123 too ambitious? Well, it intentionally is very ambitious. That's by design, because the goal is to be in a ready state for future advances in computing power and future interatomics data, such as binding affinities, kinetic reaction parameters, and so on. So what happens when all those arrive? Are we ready to immediately use it as it keeps arriving? That's our goal. As I see it, there are big psychological barriers more than any concrete barriers. For starters, being very ambitious, some people may be turned off by the long road ahead. Also, it's something of a hard to find mix of interests and skill sets. What Life123 tries to do may be too mathematical for biologists and too much biochemistry and biology for people with a computer science or math background. I continue to see firsthand the power of psychological barriers in another field, graph databases, which is one of my areas of expertise. There is a fair bit of demand for expertise in that area, but a surprisingly small supply of experts, which is great news for me in terms of easily finding jobs in that area. But why is that? There is no good reason, as far as I can tell, because graph databases are actually pretty easy compared to regular relational databases. It appears to be a psychological barrier. I can see no other reason. Graph databases seem to be the most natural thing in the world for people with background in both computer science and math, like me and a former boss at the big pharma, but presumably somewhat intimidating to many computer scientists or data engineers. I'm speculating, but the point here is that the power of psychology should never be underestimated. Another barrier is that being distracted by all the what's sexy AI and all the hot big data fields, those are taking everyone's attention. Here's a great quote, a personal communication from a researcher. There are a lot of quantitative people in bioscience, but they normally practice more data-driven informatics approaches rather than modeling. Most of them are from computer science background. Systems biology is indeed more appealing to physics majors. When you combine the social and psychological elements, a quote comes to mind, a poem about two roads that diverged in a wood and taking the one less traveled by. So this is where dynamical modeling stands as I see it. Talking about whole cell modeling, which is the main initial goal of Life123, well, it's an ambitious goal, but it's not completely new by any means. I could easily find examples in the literature from back in 2012, for example, a great article in the Cell Journal about whole cell simulations of Mycoplasma genitalium, one of the simplest bacteria. And there's a 2015 article in Trends in Cell Biology about why to build whole cell models. Well, that's exactly what we want to be able to better quantify. Virtual cells are a goal of many, and that includes Life123. With a virtual cell, we would like insight into questions such as how it will respond to inputs, or combinations of those, or inputs never seen before, or inputs while in a particular state, or what kind of inputs to provide to attain a desired result, and so on and so forth. Also, how does the cell's response vary when the cell undergoes epigenomic changes, which is very relevant to aging and health span, as well as how to emulate real-world phenotypes, which incidentally could also be used as a way to estimate the missing or poorly known parameters of the dynamical model. That brings us to exactly what about the parameters and the initial conditions? Every time there is a dynamical system, those are something that's needed. What do we do when there are unknown or poorly known? And that could be parameters like reaction rates, binding affinities, and so on. Or it could be the initial conditions, like initial concentrations and so on. Well, one approach is to use machine learning or genetic algorithms to estimate those parameters. With this approach, the counterpart of a loss function, in other words, something we're trying to optimize, could be the simulated cell's resemblance to real known phenotypes. Something to keep in mind is that we have been talking about individual virtual cells so far, but in the grand scheme of things, exciting as individual cells are, our interest as far as medicine goes is really more about tissues. So one question that could really help is, do we really need to have impeccable simulations of each and every cell? I suspect the answer is probably no, if what we really care about is their behavior in the aggregate. Unless there is a systematic error in each virtual cell simulation, I think it's conceivable that some inaccuracy in individual cells could possibly become less important when we look at the aggregate behavior. That's a situation often seen in statistics. There is much less variation in averages than in each component. To be clear, I don't have proof of this. It's a guiding intuition. Let's talk a bit more abstractly about dynamical systems and where life starts. If we have a system with just reactions, it's kind of boring. I mean, it goes to equilibrium after a while. End of story. Likewise, diffusion goes to equilibrium. But with diffusion together with reactions, some dynamics can be quite interesting. And you can find videos on YouTube about fascinating two-dimensional bubbles that change over time in carefully crafted homemade experiments. And if we also introduce compartments, we start getting some very interesting dynamical systems. But at the same time, we could say that the weather is indeed a very interesting dynamical system. It definitely is, but we probably wouldn't call it alive. So what's missing? Well, when we take all the above elements and we also bring in something that works in a digital manner or partially digital manner, analogous to digital computers, namely macromolecules, then we start moving towards a system capable of computations for complex simulations. And if that system is able to compute a model of some aspects of the real world around it, so that they can react to it, for example, as a way to preserve its own existence, then we start getting something that we could start thinking about as being life. I don't have time to elaborate further, but there's an article I wrote about this and the link in the description. Let's get more down to earth. What kind of technology are we using? These are some of the technologies being in current use and some being in anticipated usage, as well as the technologies that aren't part of Life123 but have been used in its development. ChatGPT, for example, turned out to be very adept at making good suggestions on how to deal with visualization. And Mathematica has been handy to provide some validation. For visualization, the project is currently using Plotly, which has turned out to be an excellent tool, possibly supplemented by D3 and Vue, and for network visualization, Cytoscape.js. As far as math algorithms, using the forward Euler for the systems of ordinary and partial differential equations is what is currently being done. This method is like the duct tape of solving differential equations, not the most sophisticated, but very versatile. Currently, multiple norms are being used. Norms are values computed in particular manners, and they're used to gauge the onset of instability in the solutions of the differential equations. And various heuristics are being used how to best choose variable time steps in the simulations. The system is very modular. There is an element of drop-in replacement for math modules, as well as a drop-in replacement for biochemistry or biophysics models. For example, one can start with coarser, more approximate models just to get going, and then gradually replace them with finer models as needed. The project's history is as follows. Early prototype in 2020, first beta release in early 2022, and then a long series of beta releases, the last of which was at the end of last year. It's now a series of release candidates. The terminology reflects the fact that the platform has been becoming a lot more stable and adding more features. And then, in April 2025, this project had its public debut with a talk I was invited to give at this conference. If you were to ask, can I see a Hello World minimalist example? Well, on the website life123.science, under Experiments, there's a filter you can use, and it shows you different Jupyter notebooks for different available experiments, in particular the quick start ones. Here's a very minimalistic one where we import Life123, we create a uniform compartment, we add the reaction, we set initial concentrations, and then we react over a particular time span. And afterwards, we can look at the history and plot it. So what's currently available as features of the platform? Well, reaction kinetics of a network of reactions, diffusion in 1D and 2D, including reaction diffusion, support of enzymatic reactions, compartments in 1D and 2D are imminent, and also there is an early implementation of macromolecules. Those features I talked about are really just the tip of the iceberg. So what's the bottom of the iceberg? Well, it's a battery of tools, because there's a lot that needs to be dealt with, including numerical instability, throttling up and down the computational effort, visualization, analysis of results, diagnostics, using high-power computing. Those are things we would like to have in the platform, so that individual researchers who are exploring some aspect of systems biology don't have to deal with all this. What about some challenges? Well, computing resources, for one, modeling particular elements of biochemistry and biophysics and so on, working on different scales of space and time, the estimation of the parameters and initial conditions, as discussed before, integration and coordination of efforts, staying funded and active while remaining open source. Some of the challenges. Let me wrap up with future directions. They include, at least in the short term, things like scaling up, parallelizing, a full implementation of the features I mentioned before, like compartments and macromolecules, better dealing with enzymatic reactions, given limited knowledge of their parameters, better math algorithms and heuristics, integration and comparison with other open source software. And also exploring using the Life123 platform to create a version of existing books and papers on systems biology, such as the one shown on this page, whose author, incidentally, has background in, surprise, surprise, physics, and also moving on to 3D simulations, and that element of machine learning to estimate the missing parameters, as discussed earlier, as well as community involvement from a wide variety of disciplines. On the website, there is a Participate page that includes boxes like the one shown on this slide about some current needs and skill sets to help in those areas. And anyone with that particular skill set is welcome to join in the effort. We would also like to be able to have community uploads of In-Silico experiments along the lines of Kaggle for data science. So, Julian West, that's me. I'm the founder and project lead. My background includes math, computer science, and molecular biology. I also lead another more established open source project, brainnx.org. The first scientific advisor to this project is Kevin Parrott, who is a professor of biochemistry and the owner of OpenCures, and also works in the Bach Institute on HealthSpan. On our website, there is a 3-minute intro video, also linked in the description, as well as interactive In-Silico experiments in the form of Jupyter Lab notebooks. And there is a one-click hosted version, as well as guides, microblogging, and a sign-up for community participation.